Molecular dynamics simulations. Molecular dynamics simulations are computer simulations that use numerical integration to study the behavior of atoms and molecules over time. MD simulations are computational tools used to investigate the dynamics and behavior of biological macromolecules. These simulations provide a detailed understanding of molecular interaction. The picture of molecular dynamics simulations are shown that how they take place. First, there is a starting structure, then there's the preparation of simulation system, and then there is simulation run, and then there is the analysis, trajectory analysis. Use of MD simulation in the study of biological macromolecules. Use of MD simulation in studying some of the biological macromolecules in proteins, nucleic acid, and membranes. Protein folding. MD simulation provide insights into the folding process of proteins by tracking the atomic level movements of amino acids. Conformational changes. MD simulation enables the study of conformational changes in proteins such as domain movement, loop dynamics, and protein flexibility. Protein-ligand interaction. MD simulation can predict and analyze protein-ligand interactions, aiding in drug discovery and design. Simulations can explore binding modes, binding affinities, and the dynamic behavior of ligands within the protein binding sites. Here, it is shown that how simulation takes place in protein dynamics, how protein binding, protein folding, and conformational cycling, how quantum chemistry helps in it. Nucleic acids, DNA, RNA structures and dynamics. MD simulation provide insights into the conformation, flexibility, base pair dynamics, and global structural changes of nucleic acid. DNA protein interaction. MD simulations are instrumental in understanding the mechanics of DNA protein interactions. They can explore protein binding modes, identify DNA binding sites, and analyze the dynamics of protein DNA complexes. RNA folding and function. MD simulation contribute to unraveling the folding pathways and functional dynamics of RNA molecules. Here, the structure of nucleic acid is shown that how quantum mechanics and MD simulations are involved in it. Membranes, lipid bilayer dynamics. MD simulation provides a detailed understanding of lipid bilayer properties, including fluidity, phase transition, and lipid ionization. Here, the fluidity and phase transition and lipid ionization of lipid bilayer is shown in the figure. Membrane protein. MD simulation are crucial for studying membrane proteins such as channel receptor and transporters. Simulations can investigate protein induction and folding into the lipid bilayer, explore protein dynamics, and analyze protein-lipid interaction. Membrane fusion. MD simulation contribute to the understanding of membrane fusion events such as viral fusion or vesicle fusion. Software for MD simulations are GROMACS, a widely used and highly optimized software package for molecular dynamics simulation suitable for simulating a variety of biomolecular and chemical systems. NAMD, a scalable and parallelizable software package that specializes in molecular dynamics simulations of large biomolecular systems. LAMMPS, a versatile and widely adopted molecular dynamics software package capable of simulating diverse systems, including material, polymers, and biomolecules. Key formulas and calculations involved in molecular dynamics simulations. Force calculation. The force acting on an atom is typically calculated using an intra-atomic potential or force field. The force is a negative gradient of the potential energy function with respect to the atom's position. F is equal to minus delta V R. F represent the force vector, delta is the gradient operator, R represent the position vector of the atom. Equation of motion. The equation of motion describes the evolution of the positions and velocity of atoms over time. They are typically derived from Newton's second law of motion. F is equal to ma. F represents the force acting on an atom. M is its mass and A is its acceleration. The acceleration is related to the second derivative of the position vector with respect to time. A is equal to dr square R by dt square. Integration of equations of motion to simulate the time evolution of the system. Numerical integration methods are used to update the position and velocities of the atom based on the forces. The most commonly used algorithm is the Verlet algorithm, which is finite difference approximation of the equations of motion. The Verlet algorithm uses the following formulas to update positions and velocities. R into t plus delta t is equal to R into t plus v into t delta t plus 0.5 A into t delta t squared. V into t plus delta t is equal to V into t plus 0.5 into A into t plus A into t plus delta t into delta t. Delta t is the time step used in simulation. Quantum mechanics, molecular mechanics simulations. Interaction between drug-like small molecules and large macromolecules. Quantum mechanics and molecular mechanics simulation combine quantum mechanical calculations which provide accurate description of the electronic structure and chemical bonding with molecular mechanics simulation, which approximate the larger macromolecular environment using classical force field. Description of QM region. The QM region includes the small drug-like molecules and the atoms of the macromolecule that are directly involved in the interaction of interest. The Q The QM region is treated using quantum mechanical methods which allow for the accurate modeling of electronic properties, chemical reactions, and bond formation and breaking. Description of MM region. The MM region comprises the remainder of the macromolecule, including the surrounding solvent molecules. The MM region is described using classical force fields which approximate the interaction between atoms based on empirical potential energy functions. QM-MM interaction. The QM and MM interaction regions interact through a coupling scheme that allows for the exchange of information between two regions. Typically, this is achieved through a boundary surface where the QM and MM regions meet. Applications of QMM and MM simulations. Ligand protein interaction, enzymatic reaction, solvent effects, reaction mechanism. Ligand protein interaction. By considering the electronic structure of the ligand and the protein surrounding environment, the QMMM simulations allow for more accurate representation of the interaction involved in ligand-protein binding. Enzymatic reactions. QMMM simulations are applied to investigate the mechanism of enzymatic reactions and understand the role of active site residues in catalysis. These simulations provide insights into reaction pathways, transition states, and the energetics of enzyme-catalyzed reactions. Solvent effects. QMMM simulations are valuable for studying the influence of solvent molecules on ligand-protein interaction and enzymatic reactions. The solvent environment can affect the energetics, and dynamics of the system. And QMMM simulation allow for the inclusion of explicit solvent molecules in the MM region. Reaction mechanism. By considering the quantum mechanics description of the active region and the surrounding protein environment, the simulation provide detailed insights into the reaction pathways, intermediates, and transition states involved in complex chemical transformations. Challenges and consideration. QMM simulations presents several challenges and consideration. The choice of QM region size, the selection of appropriate quantum mechanical methods, the accuracy of the force field, the treatment of boundary conditions. Key formulas and calculation involved in the quantum mechanics, molecular mechanics simulations. Quantum mechanics calculation in QMM simulation, the QM region, which typically includes the chemically active part of the system, is treated quantum mechanically. The QM calculations are based on the principle of quantum mechanics, often using methods such as density functional theory, DFT, or wave function based methods. The main QM formula used is the electronic Schrodinger equation. H psi is equal to E psi. H is the electronic Hamiltonian operator. Psi represents the wave function. E represents the energy of the system. And the equation states that the Hamiltonian acting on the wave function gives the energy of the system times the function, wave function. Molecular mechanics calculation. The MM region, which typically includes the non-reactive part of the system, is treated classically using force field. Force field approximate the potential energy of the system based on parameters such as bond length, bond angle, and non-bonding interaction. The force acting on an atom in the MM region is given by F is equal to delta V into R. F represents the force vector, delta is the gradient operator, V into R represents the potential energy function of the